we are going to start MVZ lunch on Berkeley time, even though we're doing it by Zoom this semester, just to accommodate people who have another meeting that may end at 12. So I want to welcome everyone to our first Zoom MVZ lunch. And we're going to continue like this all semester long. Normally at the start of the seminar, I would read out announcements about upcoming seminars, but since they're all being done by Zoom online, instead, Carly is going to send out the announcement by email. And so I saw that she sent that this morning. So I encourage everyone to look at it, and you can attend other seminars like Fossil Coffee or IB Seminar. I just want to make a comment about IB Seminar for all of those of you, most of us who are in IB. IB seminars are happening by Thursdays at 12:30. And I've noticed that attendance has been spotty among the students especially. I've been pleased to see that attendance among the faculty has been higher than ever. But I encourage everybody to attend IB seminars Thursday at 12:30. We have a great lineup of talks for the semester. So with that, I'm going to turn it over to Becca, who's going to introduce today's speaker. But the one thing I would say is I'd like to ask everybody to mute themselves and questions in the chat. So Rebecca, take it away.
Thanks, Michael. I'm pleased to introduce Jaime Chaves, who just started as an assistant professor at San Francisco State University. So he's new and local. And I want to thank him for agreeing to do this virtual format. So Jaime is not actually new to the Bay Area. He was here at SFSU to do his master's in 2002. Then he did his PhD, finishing up at UCLA in 2010. And since then, he's been working in Ecuador. So he's also an adjunct professor. I think it's USFQ. Is that right, Jaime? Didn't write that one down.
Yeah, you're right.
Great. So done a bunch of work on the Galapagos, which I know we're all going to be jealous of, working with the classic finch system. And I'd also like to share that Jaime is an awesome artist. So if you go to his website, he's done some beautiful paintings of birds and also street art in the Galapagos. So thank you again, Jaime, and I'll let you take it away.
Perfect. Thank you so much, Becca, for the invitation. This is, as you mentioned, it's great to be back in the Bay Area, and what a great way to come back by sharing some of the work that I have been, interested in the last 15 years as a postdoc and as an adjunct professor in Ecuador, and now back in San Francisco State. And so what I want to share today is a little of those major questions that I have trying to tackle using this iconic system of birds from the Galapagos, the Darwin finches. But to give a little bit perspective of the history of these finches, this group of birds, we get to know after this incredible voyage that Charles Darwin was on board of the Beagle. And only a couple of weeks ago, this trip happened 185 years ago. And when Darwin got to the Galapagos, he was collecting lava rocks and shells. And some of the birds that really, caught his attention were mockingbirds. And as you can see here, a photo of the birds that he collected. And they really help, start this notion of different species with different morphology on different islands. So the finches were not really the group of birds that got his attention, at least originally. So it's kind of ironic that these birds, the finches, are being named after Darwin, when these, mockingbirds would have been called, or should have been called, Darwin's muses or Darwin's mockingbirds. But the Galapagos finches were called this way before we actually know as Darwin finches. So in this little chronological chart, you can see here a point of inflection when we do start calling them Darwin finches and when they were called Galapagos finches. So Galapagos finches were known from the time that these birds were brought back into England. And it was in the 30s that work by primarily Percy Lowe that started using the word, name them Darwin finches. And so it's kind of ironic now that, again, these birds are named after Darwin, but it's David's lack of work, seminal work that actually used this group of birds to really illustrate the most important tenets on evolutionary theory, proposed by Darwin. And now there are examples of, the best examples of character displacement, evolution via natural selection, and obviously adaptive radiation. So, and in the words of Darwin, after he looked into this group of birds, this is the origination or the diversification of lineages that descend from a common ancestor, in which these different lineages have taken different ends. And the reason or the motor behind this diversification has been one particular trait, this adaptive trait, which is the beak. So you can imagine these populations moving to different environments. They have very different seed species also there. And these birds going through natural selection and changing by small changes at the beginning to the adaptation of new seeds in these different islands that, although they're quite near from each other, they possess very different environmental conditions. So this type of movie or a story about speciation has been beautifully captured in one of the books that to me really captivated my mind and in a way prompted me to study evolution. And this is the book written by Jonathan Weiner, The Beak of the Finch, that gives all these previous information and how, in particular, two scientists, Peter and Rosemary Grant, by going year after year to this small rock island that you see on the bottom left there, where they capture every single bird on this island and band them. And by coming every year afterwards, they could see which birds survived and which birds did not survive. And also, what were the environmental conditions that were shaping or affecting the survival of some of the types of birds that they were seen on the islands. So they pretty much recorded evolution in action. And this information has really put forward, the way that we see the effect of environment in shaping dramatic changes in morphology. So in these charts here, you can see the effect of environmental and climatic changes like El Nino year, a very wet year, and how that affects the amount of seeds or the type of seeds that are present on an island. And by these dramatic changes in resources, birds trace back or trade back the amount of food resources and adapting and obviously getting rid of the individuals that might not possess the type beak or the type size of beak. They're able to crack those hard seeds, or the smaller beaks that are able to maneuver the seeds properly. And in this graph here on the top right, you can see the dramatic changes in beak size from only a couple of years between a very wet year to a very dry year. And that, obviously results in this adaptation through every single generation. So one of the things that we now understand and know very well is that these birds have a tight association with the type of food that are available on different islands. So one of the major questions that are now opening up is, well, what are the consequences of possessing these different beaks that are shaped by natural selection and that have been modified for different ends? And here's where I'm going to try to cover these four aspects of my research, in which each one of them have different stories about how the study of beak size and beak shape could inform us about some of the consequences that these birds might have through the process of natural selection. And I'm going to start with looking at the beak genes, where are the building blocks of these important tool. Talk a little bit about thermoregulation and how beaks are actually being used for transfer of heat, and a little bit about microbiome, and finally about urbanization on the Galapagos Islands. So let's start with the first part of the talk. I'm going to be covering big genes. And if some of you have been to the Galapagos, and you could probably be, facing this conundrum of, and if you're a bird watcher, even worse, trying to come up with and skill to identify these finches. So if I ask the audience right now, if you could probably come up with a number of species based on this photo, how many species you think you see in front of you right now? And, I think it's easier, maybe just do the two extremes. We know, okay, we can have, a small bird there on the left top, and the one large one in the bottom right, and maybe one in between. So you can say probably 3 species. But if you know a little bit about the work that Peter and Rosemary Grant have been doing, this is the same species from Daphne Island. So these are all the different variants of the medium ground finch at different time scales and different time frames in their history. And if you throw in there the 2 extreme species, the small ground finch on the left and the large ground finch on the right, what you have in front of you is a morphological continuum. And it's going to be very hard for anybody, even very skilled ornithologists, to try to come up with where do you draw the line between these species. So given this incredible variation in beak shape and size, one of the questions that I wanted to look at is, well, can we tell any way that we can discern these morphotypes based on genetics? And what are the building blocks of this incredible variation? So the first question for the talk was, which genes control beak size variation? Now, one of the advantages of working with a group of birds that are highly and very related to each other, and is also kind of a young radiation in terms of geological time, around 1 million year old, is that most of the genome is going to be very similar. But if we're going to try to pinpoint regions in the genome that are variant between the 2 or the 3 morphotypes, maybe we can pinpoint those easily. And in order to do that, using this type of genome-wide association analysis via rat sequencing, where we chop all the genome using restriction enzymes, and by comparing pairwise comparison between individuals that differ dramatically in beak size, one could go around trying to figure out what part of the genomes are more divergent between birds that look very different. And in this case here in this graph, if you're not familiar with Manhattan plots, which is going to be like the skyline of the pairwise comparisons of these regions, any 2 species that differ dramatically on morphology and that is associated with a particular region of the genome, they're going to come up above this dashed line here on the graph. And on the y-axis, you have the position where these particular fragments of the genomes are located. We have genome annotations, so we can know which part, at least in which chromosome they were. So it was not surprised that most of the dots that you see here, which is every single one of those genetic comparisons, are found below the dashed line. That is, that it's almost impossible to tell apart any type of the 2 morphotypes based on most of the genes. Actually, only 0.003% of the genome differs between some of these large, medium, and small birds. And the interesting thing is that these particular regions, they explain over 80% of the beak variation that you do with doing this analysis. And in particular, we can see 2 regions, or at least markers, that we were able to identify. These were DLK1 and HMGA2, which are genes known now that are related with morphogenesis and cartilage formation in an array of species. So it was at least very interesting to know that there are large effect genes that are controlling for beak variation in size. But we also wanted to look at what is the variation of these genes across all these different sizes of beak birds. So in order to do that, we genotype, in this case, I'm presenting just the data for HGM2, and look at the variants and the alleles that you can find on each one of these different morphotypes. And what we found is that for every single small bird or small ground finch, they all have or they possess the GG allele for that particular gene. And the opposite was true if you are a large bird, you have the other allele for that one. Now, within the medium ground finches, we found an array of information in which the small version of the medium possess the same allele frequency or allele variant as a small ground finch. The large medium has the same AA, and the intermediates were heterozygotes for this gene. So the interesting thing is that all this allele variation is maintained widely in these populations, which is, makes sense. This is the fabric for natural selection. So by keeping all these variants in the population, you now can have all these incredible different groups of birds that might show up at different times of history that are shaped by the environment. So we're taking advantage of these type of analysis in which we have very well sampled genomes of these birds. And we're trying to come up with other type of comparisons. So using this GWAS analysis, we now know that there's so many other differences in behavior that these birds exhibit, taking advantage that these are young gradations. So now we have full genomes to make more thorough comparison between those. And as I mentioned earlier, the differences that you can find between these 2 or 3 species are so dramatic that most likely any type of gene association that you can find might pinpoint to the right part of the genome that might be under selection that would have allowed you to understand this dramatic diversification of these birds. But you know that beaks are not just used to crack seeds. And beaks have been heavily studied in thermoregulation, and in particular in the way that beaks are used to dissipate heat. So in this paper by my co-author and colleague, Glenn, he looked into the role of the large beak of a toucan and how a toucan is used as a heat radiator during the hot day or the hot part of the day in which blood is being flow into this large structure to have an exchange of heat with the environment. And when it's cold, you can see here on the bottom part of the photo how the contraction of these vessels now prevent heat to get lost in the environment. So this is one of the ways birds thermoregulate with their environment. So given this tight association between beak size, we thought maybe it would be interesting to look at what is the effect of these variation in beak morphology and how are birds adapting to a very hot environment like the Galapagos Islands, where you also have a very dramatic gradient in temperature if you go up and down the elevational gradient. So we went into looking at how does beak size influence thermoregulations in Darwin's finches. So one of the things to get this type of data is that you either have to have the bird close to you, and going around with the spatial lens on the Galapagos was kind of tricky. So we were able to build this oven bird, for the purposes of this work, and we were able to put the bird inside this microwave and crank up the temperature and start getting information about what part of the body would start dissipating heat first. But we also were able to collect information about the temperature of the environment, obviously gas, CO2, water loss, and evapotranspiration. So all these variables that were going to be correlated to the heat. And obviously when, when you go too high and start smelling like Kentucky Fried Chicken, we just, turn the dial down and get the bird out to get it out so we don't get too hot in there. And one of the things that we find is that with expectation, the larger beaks, they obviously have a high evapotranspiration at higher temperature. So you have a big bird that is going to lose a lot of heat very quickly when the temperature rises. And so that was not really something that went actually correlated very well with literature. But one of the things that we also did is that we followed these birds after the essay. And one of the things that you can see is that the temperature in the environment really affects the activity of these birds. So birds that have very long beaks, they're usually active during the cold part of the day because of these threshold in which you're going to lose too much energy trying to thermoregulate on a hot day. So larger big birds are active early in the morning, and as the day moves on and warms up, the other species are more active. And in a way, it's interesting because you can now see that there's, in a way, the species are not interacting with each other. There's little less competition. So we think now that this could be another axis of selection of this physiological displacement. So birds are not competing for the same resources at different times of the day, because there's a pressure on the energy that has to be expended in thermoregulating. The other interesting thing that we found is mentioned that we have birds that are found across an elevation gradient. And then we have a point in which the bird is about to enter thermal stress. And that's the time where the birds start gaping, which is opening the beak. And you can actually see in the camera the amount of vapor that comes out of their beak when they're like at the extreme of it. And we were able to get to those thermal tolerances. In lowland birds, that limit is quite high compared to birds in the highlands. So birds in the highlands, they reach the tolerance very, very early on. And this is something that we're going to start exploring later on, because it seems that there is at least a difference in the way the birds are adapted in the lowlands, where it's more hot, to withstand hotter temperatures. And this is going to be a work that we're going to be doing in combination with folks at UCLA that has to do with this mismatch between genes that are usually found in birds that are adapted to environments that are warming and which are surviving better, that compared to birds that are suffering from these wintering grounds, when they might be a mismatch between the genes found for heat tolerance. So this is the work done by the project called Genoscape. And in that way, you can start mapping genetically, where do you find this genetic vulnerability? Do species have what it takes to survive warming scenarios? And by making predictions in the future about areas that might be prone to warming, you can start identifying what population might be sensitive to climate change, which species or what populations might make it if they do have the genetic tools to do that. And there's any way we can help out in a way to these species. Some work that's been done with corals, for instance, they're doing trying to translocate or do gene flow experiments to see if at least part of the genes can be moved and prepare these species to withstand climate change. Now, obviously, birds are using all these different resources, and something that we know a lot about microbiome, and particularly gut microbiome, is that there is a very tight association between the food items ingested by animals and obviously humans, and the type of microbiome that you have in our gut. So this tight correlation between the microbiome, gut microbiome identity, different bacteria that we find, is something that is also known to be the first line of defense against a lot of pathogens. And was one quote by Lee et al. That really caught me thinking about this, in which you can envision in the process of diversification of these group of birds taking on different trajectories, and particularly taking trajectories that are driven by food, that you expect that somehow the same divergence that you will find between the host lineages should be mirrored by their microbiome, knowing that there's a tight association between those two. And with finches, knowing that they are dramatically divergent in the type of food that they feed on, in the case of the seed eaters on the top left and top center, to the vegetarian finch on the top right there, the woodpecker finch that feeds on arthropods using opuntia, cacti, little spines, to the cactus finch feeds on some nectar and pollen of the flowers of the opuntia, to the vampire finch on the top right. This is, again, a very interesting group to look into these type of associations, with the expectation that if you are going to sample the genetic diversity of bacteria found inside of these birds, you can come up with a hypothesis about how is actually a diet affecting finch gut microbiome. And the way the microbiome work is done, to at least to identify the amount of diversity that we found on the gut microbiome, It's based on genetic similarities or distance. So you can envision in this, going into this research, in the two axes of divergence, that birds that feed on the same item, let's say blood, they should probably have their bacteria that are unique to that group, and you will probably find and expect to see a cloud of individuals in one part of the chart, versus the group of birds that feed on arthropods, and they might be in another part of the chart, and so on and so forth, in which you have all these clouds of the different species of birds associated with their own unique set of microbiome, at least bacteria. To our surprise, we did not find any significant difference between most of these species when we exclude one group of birds, which are the vampires. When we include the vampire finches in this comparison, there is a significant difference between those birds and the rest of the samples that we collected. So there's something interesting happening, at least partially, within the vampire finches that is not the same across all these other birds. So those birds are eating very different things, but it doesn't seem to be a significant difference between the gut microbiome composition that you find on these birds. So what are these vampire finches? This is the only bird in the world that inflict a wound to directly benefit from blood of their hosts. Now, we know that there are other species in Africa that also feed on blood, and many other birds that feed on blood, but they are not directly inflicting a wound on these birds. In this case, this is a masked booby on the island of Wolf. And these birds here, this is a learned behavior. So birds here, you can see some of them are just checking out around because they need to learn how to do that in order to survive for half of the year, because half of the year is so dry that there's nothing really out there to feed on. So these finches rely on this type of behavior to obtain nutrients and water from the one of the largest colonies of masked boobies on the Galapagos. So with this type of information, we wanted to identify, so what is so unique and different on these birds that is making our comparisons so different? So one interesting thing is that we found that the outlier here is a group of bacteria called Fusobacteria. And this is a bacteria that is also found in carrion-eating birds and carnivorous birds. So now you have this small passerine in the middle of the Pacific that has the exact same gut microbiome that a vulture or an eagle would have, that have very different evolutionary histories. Now, you can think about these in terms of convergent evolution to these different feeding diets. And this is something that we wanted to compare to other vertebrates that are known to feed on blood. And if this is the case, we compare with the gut microbiome of bats. And we wanted to look at if there was any convergence either on their taxonomy, that is the species identity of the bacteria, and on the function that these bacteria might have, breaking down some of the elements that are found on blood, which are amino acid, heme and iron elements, fatty acid chains, and sodium. So here in this graph, we are comparing birds within the finches that are hematophagous, yes and no on the bottom, and also between bats that are not and yes for blood feeding. So I would just get your attention on the right column for the comparison between the finches and the bats in order to look at what type of bacteria are found between finches and bats. And the first thing that we found is that there seems to be quite convergence, at least with a group of bacteria called the Peptostreptococcaceae, which are known to produce genes, proteins that allows the digestion of iron and the transport of sodium. So there seems to be a partial convergence, at least in this big category between the vampire finches and vampire bats. But as you mentioned from a previous slide, Fusobacteria here in orange is prevalent in vampire finches, but it's not found on bats. And we now know that Fusobacteria, at least on birds, is important for the production of vitamin B12. And this could be something that these birds are in need. And by having this unique adaptation, they can be able to metabolize some of these elements in the blood. So, okay, so there's partial convergence in taxonomy. What about the function of the genes? Because it doesn't matter if you have different species of bacteria, but if they are doing the same thing, you might be able to have quite a convergence, at least in the function. And in order to do that, we assay the genes that are known in the same categories for amino acid, metabolites, heme, sodium, and urea. And by comparing the different genes found on these K lists, these are the name of the genes that are found on this orthology database, we were able to find that there is quite a lot of genes that are of known function that are shared between the two different species of vertebrates on different species of bacteria. So there's a similitude on the function of the genes that are contained in the microbiome of these two vertebrates that might allow them to be successful on digesting blood, which is one of the most common elements, but very hard to digest and use the nutrients there. So how do you actually go around looking at other type of associations for diet is that you can use isotopes for this. And the two most commonly found isotopes in nature are carbon-13 and nitrogen-15. And depending on the ratio that you can get from these, from the skin or from the feathers of these species, in general, you can get a sense of the type of diet that are associated to. So in the case of, we wanted to look at, the Darwin finches that feed on blood compared to other species. And here we just compare across a lot of data sets. And you can see that obviously vegetarian and ground finches have the lowest ratio of these two isotopes. And as you move up in the chain, you have obviously your big carnivores that have high numbers of carbon-13 and nitrogen-15. And we found that the finches that feed on blood has one of the highest nitrogen-15 values. And we think here is that these birds are stealing the isotopic signature of the marine birds that feed directly on fish. And by stealing that isotopic value, they are incorporated in their feathers. That's the reason why we are able to get this super high value on these birds. So one of the things that we're now trying to explore with this research is what is the origin of fusobacteria, why and how these bacteria made it to the Galapagos, and is only found on these vampire finches and is found in nowhere, in not other species in the Galapagos. So we have a couple of hints there that we're going to start exploring. And also, as I mentioned, this is a behavior that is learned. So the birds are born without this information. So we try to figure out, is there any way that we can analyze and study this behavioral component? And the other thing that we're doing right now is that we're getting saliva proteomics to look at to which extent there's any proteins in their saliva, like in bats that have a potential for this adaptation. And that way we're going to be comparing another level for convergent evolution to blood feeding. Now, one thing that you will know if you get to go to the Galapagos is that the Galapagos are really not that isolated anymore as they used to be. And one of the things that has been very pervasive now in the Galapagos is that this is a place that sees, well, not now, but it sees around 350,000 tourists every year. And there is a huge impact in the way urbanizations and anthropogenic activities have influenced the evolution of birds. And urban habitat, they have created this unique opportunity to look at how birds might be responding to these new environments. So we again have a system in which the settlements on these 4 islands in red here encompasses around 40,000 local people, residents there, plus those 350,000 visitors a year. It really is putting the Galapagos in a new trajectory in which we now see the interplay of these new forces with the response of birds in these islands. So one of the things that is very common is that birds have now easily adapted to live in the city. If you're having your breakfast in the morning, this is the plantain bolon, these finches will have no problem in trying to steal it from you. You can see them all over the town eating chips and crisps and popcorn that you see here. So this is the normal scenario. And this is a video of a friend of mine, which a grandmother has been providing this additional food for finches for over 25 years. So she's pretty much putting up one of the most incredible experiments out there to see to which extent these, the effect of human food might have on finches. And knowing the history of this close association of food items and the evolution of the birds, it makes sense here to really start exploring the impact of human activities on these islands. So we wanted to explore how the finches respond to novel food sources, particularly food introduced by human. So one of the things that we wanted to look at is there any preference that these birds have already developed? Because one thing is, okay, yeah, well, they're eating because they're in the city. But what happens if these birds are being provided these same items somewhere else? So what we did here is this cafeteria experiment in which we put a tray with human food. So you have crisps, biscuits, and rice. But also, we presented with the seeds that are usually found in their habitats and are known for these birds to go after. And these same experiments were replicated in the town in different spots, and also in parts and areas where they're far away, that there's no human intervention. And we wanted to compare to which extent birds are used or not, or have a preference for human food and or their natural food it presented equally. So the first thing that we found is that when you go to these remote places where most of our research takes place, birds really don't get much. They don't even care about this item being placed in the middle of the field. We have very little visitations, and we cannot find any significant difference between any of the food items that presented to the birds. But when you compare these to the experiments done in town, you can see that birds, given the option between their human and their natural food, they preferentially go for food that are related to humans. In particular, they prefer quite a lot the biscuits and the crisps. So this obviously is something that is being developed, and there's a preference that these birds have been being passed on. So we're trying to figure out what are the consequences of these preferences. What is going to happen to these birds if they are eating items that have very different selection pressures that they would the seeds that are naturally presented with? And one of the things that we know is that we might be looking into species that are changing dramatically the beak. And this is a very good cartoon in which one of the finches asks their mom why his beak looks different than his brother, and the mom is worried that he will ask it one day. It's a secret, but your brother is adapted. So there seems to be quite a difference and effect of the adaptation of these birds quite rapidly by the introduction of these novel pressures in the islands. And one of the most dramatic work has been done by our collaborator, Andrew Hendry, by looking and using collection data, looking at how the beak looked before humans on these islands, and the beak of some of these finches that are now in town, have there been any difference or not? So one of the things that Andrew found out is that there's been a loss of this bimodality. So that is that you have these populations that are tending towards two different opposites in beak sizes. So there might be two different types of seeds that are already shaping this distribution. But what happens when the human population move in and change the selection pressures? Now intermediates that were previously selected upon in years prior now are doing much better. And we think that these are the birds that are now taking advantage of these soft seeds or this soft food that is not presented by humans, and eroding, or at least deleting, erasing some of this bimodality that was present originally in this area. But we also know, and linking a little bit about these associations between food preferences and microbiome, we can now also try to explore, so what are the consequences in terms of the gut microbiome that you find on birds that are now living in the city or in the town compared to birds that are elsewhere? So we did this experiment as well. We collected the data from these different birds, and is no surprise that we find that at least the microbiome diversity is much lower in birds that are found inside town. And this probably has a lot of consequences, knowing the tight relationship between microbiomes and health. So as part of this collaboration, we're now researching links between microbiomes and diseases. There are many diseases that have been introduced to the Galapagos. Some populations seem to be more immune to other ones. We're trying to find out if there's any relation between other type of elements like parasitic flies that are laying their eggs inside the nest of these birds, and there might be a correlation between also microbiome diversity and which birds are able to cope with this parasitism. We're looking at health parameters and nutritional physiology. We know that some of these items might not be great for their health. And looking at the genetic implications of these by looking at telomere shortening and some oxidative stress, looking at hormones that has to do with the differences between the food items present in town compared to the more natural setting. So to wrap up quickly here, a little bit about the talk. We now know, and we've always been, been presented with this group of birds that are so iconic in illustrating this mechanism of natural selection, shaping species formation. And it is one of the best examples for adaptive radiation, in which the combination of isolation, time, and these extreme diets are really the engine in the proliferation of species. And we have, show that climate, in particular temperature, could be another axis of selection that we previously didn't know exactly how was this playing out. So maybe now you have an interplay between food selection and temperature that might be influencing some of the bird coexistence in different islands. But also they are telling us something rather dramatic about the way we are changing the planet, and how these birds, luckily at least, these birds have this capacity for very quick adaptation. And they're telling us something that we need to do something about, because now these pressures that we're putting in the planet, at least for these birds, they're able to adapt, but there's going to be so many other species that might not have the time or the evolutionary capacity to withstand some changes. So in a way, they're telling a story about the impact of humans in the planet, and something that we all should be aware about. So to close up, I just want to thank, obviously, all of you for being here, and Rebecca for the invitation. But there's a lot of people that have come and to the field with me and allow us to collaborate in so many different ways. All these different, this voyage that I took you from physiology, from microbiome, it would never be possible as a one-man show. So there's so many important collaborators, and really support this interactive and interdisciplinary collaborations. Without further ado, obviously Galapagos National Park and Galapagos Science Center and Galapagos are the major facilitators for most of the work that I do there. Thank you very much, and I'll be more than happy to take any questions.
Thanks, Jaime. I'd like to request everyone to just unmute themselves for a second, and let's try to give Jaime a realistic sound of applause. And I'm happy to facilitate questions if people want to either raise their hand or type questions into the chat. I can start us off though. So first of all, I didn't know about the vampire finch. What an incredible animal. That is just really, really neat. And I had some question, I had to question if they had the same sort of painkiller stuff, but you kind of talked about that.
Well, so that's the thing, we still don't know. We're just getting the proteomic data back. The cool thing with bats is that they do have a numbing agent protein as well as an anticoagulant. Mm-hmm. And the name of the protein cannot be even better, called draculin. And we tried, that's one of the questions, is that to which extent you see this convergence also at the level of the saliva. Now, I have to say that a bat does the job much more elegantly. You barely see a mammal, complaining by having a bat on trying to, get their blood. These animals are quite brutal. I mean, you see the birds trying to just like flush away the bird. I mean, they know something is going on. I'm sure you can see that there's pain involved. Mm-hmm. So I don't think the saliva is doing anything there. They just aggressive.
I can't believe the birds put up with that. You think they fly away?
Well, they do. They will, after a few minutes, they will fly away, but they have to come back, right? And
yeah, because they have the colony there.
Yeah.
Huh. I see. Okay, I see that Maho has a question.
Hola, Jaime. It's
Hola, ¿qué
great to
tal?
see you. So I have a question regarding the hogs. So I wonder, like, since the hogs are also eating sort of like blood, more or less, if they are also have some like microbiome or like related um, things that are allowing them to eat this like blood and meat.
Yeah, I think that's a great question, and that's an open, open question for anyone to go and explore that. But you're right. I mean, this is the only, besides the short-eared owl and the barn owl, with the Galapagos hogs, there are 3 raptors that you could find on the islands. But Fusobacteria was not found on any other of the finches that we find in there. And I just had to give a little hint about where we're trying to look at right now in terms of where this Fusobacteria might have originated, is that we know that these big masked boobies, they are covered with feather lice and ectoparasites. There are blood obligatory, so they feed on blood of these birds. And one of the work done by Robert Bowman, who actually was a professor at San Francisco State, and spent time on these islands looking at these same biparent finches, which is another crazy, full circle for me here, is that one of the hypotheses for the origin of this behavior is that these birds can see picking up little ticks and other ectoparasites, not only on birds, but on marine iguanas and on marine sea lions. So it might be in this type of behavior and picking up these ectoparasites that maybe a wound was created in that event, or they were already encountered blood in there. And by eating these little ectoparasites, these parasites may have microbiomes that are being adapted for blood feeding. So in a way, they could be inoculating themselves by eating the ectoparasites, obtaining high levels of Fusobacteria that now opens up this complete new trajectory for the adaptation for blood feeding. So luckily, I was able to come here in the summer back in the States with my little vials with exoparasites that I collected from this bird. So I'm looking forward to start looking at the microbiome of these little ectoparasites and maybe trying to find the key to add up more information about this incredible behavior.
Thanks, wonderful.
Thank you.
Yeah, that's fascinating. I had another question. So at the beginning of your talk, you mentioned, I forget which gene it was, but a gene apparently associated with beak size, where you're able to actually connect a SNP genotype to the beak size. Have you been able to follow up on that? Is that a regulatory region, or what exactly is the SNP doing?
These are SNPs that are in the neighborhood of these known genes, DLK1 and HDMG2. We only know, for instance, with HDMG2 is a gene that is also found in humans and determine the height of humans. So this is how far we've gotten in terms of identifying what these genes might be doing. Now we know that the allometric changes on these birds. Because one thing is to have one beak that makes, one gene that makes your beak large, but that doesn't make your body to be small or big. So we think that there's a lot of, tight linkage between some of these large effect big genes with other genes of large effect. So by having one of the genes, you also end up with these other components that make a medium bird to actually look medium size as well. And a small bird also have not only small beak, but also small. So these allometric changes, we think there's a network of very important genes that are interacting and making that. But we haven't not explored any further on those particular genes that we found.
I'm not seeing any other hands. So if I'm missing someone, put your name in the chat. Oh, okay, here's one. Bruce, do you want to speak up? Bruce Baldwin.
In the last example about urbanization, to what extent is that examining variation within a species as opposed to possibly consequence of hybridization? It's very reminiscent of the sort of situation where you get hybridization in plants, where you have this hybridization of the habitat, in this case, the food source being very, permissive?
Yeah, I think it's, yeah, of course. We now know, I mean, we already know that these birds are, they're quite promiscuous. There's so much hybridization happening between many of these forms. I think that the important point here is this example with the medium ground finch, in which within this group, you have two morphs already that you can go in places where there's no humans, and you still find this bimodality. So a large morph and a small morph of the medium ground finches. And in a way, there's less integration, or at least hybridization between them. The females of one morph tend to prefer males from the same morph than the other one. But if you go into town, these two bimodal distributions of big morphology or big shape are now being united by this bridge of intermediates. So now hybridization might be even more easy and possible between these two morphs if you have intermediates that are actually doing much better than birds in the two extremes. So there's dire consequences about losing this type of variation in places that you can easily compare between a town and places where you don't have humans in there. So this is again another of the the consequences of urbanization in the future, for sure.
Thanks a lot.
Mm-hmm. I see that Jack has a question about the fusobacteria.
Go for it. I can't see the question. Maybe.
Cool. Fusobacteria also come from the boobies themselves, since they are meat eaters. They fish, and finches could pick up bacteria from feathers, which they bird preen, and might pick up bacteria during preening, or from defecating in small amounts of feces bacteria getting onto their rumps. Yes, this is another good source for exploration. Finches, when they are present with this, the lack of food during half of the year, they go after everything, including the feces of the boobies. So they are scavengers and they eat anything they can. And we also have fecal samples from the boobies. So that will be another comparison to look at. But yeah, you're right. I mean, right now the question is open, right? What is the origin of fusobacteria and why it's so important for these birds? So any type of hypothesis is now open for us to explore. But this is a good direction to go as well.
I think we have time for another question if anyone else has something they'd like to ask. I have to say, Jaime, you made me miss bolognese quite a lot.
The plantain, yeah, I know. There's nothing good like a good bouillon with a nice cup of coffee.
I know.
Before head into the field.
If no one has had these, they're basically double fried plantains mashed into a ball and stuffed with cheese. Yeah. It's very good.
And you're not vegetarian, you can ask the one that has pork skin chicharron also in it. Mm-hmm. It's just another level.
It's making me drool. Well, wonderful, Jaime. It was nice to see you, and thank you again for giving such an interesting seminar.
Thank you so much for the invitation. Had a good time.
Wonderful. All right. See you all.
Bye, everyone. Thank you again.